Hello, we're going through my notes for life science and cell biology in a nutshell. So we're going over cell division and the changes that chromosomes go through and cells go through um, when they're dividing. Ruben, you don't want to help me? Well, again, prophase, the chromosomes condense, they become visible. Spindle fibers emerge from centrosomes. So these are the protein structures surrounding the nucleus or around the nucleus. The nuclear envelope begins to break down. Centrosomes move toward opposite poles. Centrosomes consist of centrioles, subunits. Chromosomes continue to condense, and kinetochores appear at the centromeres. So the kinetochores are the proteins on the actual chromosomes themselves. So they bind the protein fibers of the spindle fibers, which are the centromeres. They bind, and this is how the chromosomes connect and line up on the spindle fiber. Mitotic spindle microtubules attach to kinetochores on the chromosomes themselves, and they're lined up at the metaphase plate. At the stage of metaphase, each sister chromatid is attached to the spindle fiber originating from opposite poles. In the next stage of anaphase, the centromeres split in two. Then the sister chromatids are split apart, and they're now called chromosomes. Individual are pulled apart toward opposite poles. Certain spindle fibers begin to elongate the cell. In the final phase of division, chromosomes arrive at opposite poles. They begin to decondense. The nuclear envelope material surrounds each set of chromosomes. At each of the poles, the mitotic spindle breaks down, and the fibers continue to push the poles apart until the cell finally undergoes cytokinesis, and the divides or splits up into two cells. The cytoplasm splits. And in contrast, sexual division is similar. The phases are pretty much similar in the process. So the offspring of the organism differs from the parent through recombination of the cellular materials as passed down. So two cells combine, yielding a third cell containing a unique combination of characteristics from each initial cell. So the only difference is the numbers of the chromosomes. In meiotic or sexual replication, the daughter cell receives half of the DNA from each parent cell. So instead of doubling it, you're halving the number of genetic material. So then each of the two halves from each of the parents combines into a full set of chromosomes in the zygote. Therefore, you end up with a complete set of DNA. So two half sets leads to one full set. In summary, chromosomes are rod-shaped structures in the nucleus of each cell carrying genes. Reproductive cells have half the number of chromosomes of a parent cell's chromosomes. The half sets from each of the parent cells yield a full set in the offspring. So For example, in humans, 23 chromosomes are acquired from each parent, equaling 46 chromosomes in each of the cells of a normal organism. Just a side note, algal cells are eukaryotic, contain 3 types of double membrane bound organelles. They contain a chloroplast like plants, mitochondria, and a nucleus. In most algal cells, there's only a single nucleus, although some cells are multinucleate. In addition, some algae are I don't know what that means, siphonaceous, meaning that many nuclei are not separated by cell walls. Siphonaceous. The nucleus contains most of the genetic material or DNA, deoxyribonucleic acid. In most algae, the molecules of DNA exist as linear strands. So this is general information. They're condensed into obvious chromosomes only at the time of nuclear division or mitosis. This is in common with other cells as well. So here is shown a cell and all of its internal organelles and structures. 3D showing a eukaryotic nucleus, so membrane-bound nucleus. And you have all the other things, mitochondria, maybe lysosomes. These are endoplasmic reticulum. Further away, you have the Golgi apparatus, etc. Looks very cool. Again, showing 3D eukaryotic nucleus and its organelles. Very nice graphic. Obviously, the nucleus is the more prominent part of the cell. So, more specifically in genetics, the DNA, it consists of the following. One chromosome can contain thousands of genes on a single long molecule. These molecules are genetic codes that code for proteins in cells. So whatever DNA is passed on is going to code for whatever proteins that the DNA and the genes code for in all the offspring as well. So deoxyribonucleic acid is DNA. The molecule is shaped like a spiral ladder. The sides, like on the sides, are the sugar and phosphate subunits in a chain. So it's called deoxyribose. And the rungs that go horizontally versus sugar and phosphate that go vertically, they're made of nitrogen bases called adenine, guanine, thymine, and cytosine. Adenine always pairs with thymine, while cytosine always pairs with guanine in the DNA molecule. Thymines that pair with adenines, cytosine pair with guanines, so thymine and cytosine are in the pyrimidines group, and the adenine and the guanines are in the purines group. So they link up, making a look like a ladder, like a runged ladder. And the way the molecules link up, there's a helix, and it looks like it's kind of kinked. There's a minor groove and a major groove based on the way that the molecules link in the chain. And this is an observable molecule. The atoms are coded here. and you can see the base pairs